Good afternoon to Dr. Gerald for giving this opportunity. So I'm going to talk about the genetics of glaucoma. I think all of you, clinician, concerned with the farm. Now we are dealing with the primary open angle glaucoma, congenital glaucoma, as well as the angular cluster glaucoma and pseudo exfoliation. So concerned with I am looking into how the occurrence of the glaucoma. So I think all of you know the pressure, the eye pressure should be maintained. The oversection of the aqueous humor, and then should go in and out of the eye to normalize the pressure if there is any blockage. If there, matrix that is called in the literature is a extracellular matrix. Those matrix, it is blocked the meshwork because of the meshwork. Blockage. It is increased the intraocular pressure. So that's what we studied in gene expression level what exactly going on in Indian population concerned with the primary open angle glaucoma. So this is I think I'm going to talk about there is a very recent literature, the review published in Duke University by Rand Allingham. He is one of the well-known glaucoma specialists. And concerned with that review, right now, the people extensively studied in long back. Once the myocilin is identified, that is the only candidate gene for glaucoma. They thought like that, and then they jumped. I think we identified the gene for glaucoma. But the scenario is changed. More than 16 years, there are more than 25 loci identified, and then only four candidate genes identified. It means it is like a complex disease. So it is very, very difficult to understand. Many factors involved. It seems to be the glaucoma is a very complex disease. So then they collected a well-characterized sample from different ethnic groups, and then they did the genome-wide association studies for the different ethnic population. And then they identified, that's what in the review says, so there are so many, in addition to the candidate genes, right now, this is the very current information. So using the GWAS, they identified these are the novel genes identified, related to the glaucoma in addition to the myocilin, optineurin, and W36. So see here. So I think the CAL1, CAL2, DMCO1, this particular one, SIX6, these are the genes right now is associated with, they proved functionally also it is associated with the primary open angle glaucoma. And then the TPK1, this is TPK1 exclusively the normal tension glaucoma. I think all of you know, consistent with in Indian population or in our, at least in the Arabic, so we can't be able to identify the normal tension glaucoma patients because you have to measure many times, and then it's very difficult to whether the patient is exactly normal or open angle is a problem. So here, I think if you concern with the other ethnic groups, in Taiwan and China, I think definitely they are positive for normal tension. And then the copy number variation, there are more copy number. And Dr. John Fingert from University of Iowa, in a multi-generation family, he proved this the TPK1 gene associated with normal tension glaucoma. Okay, so I think that's what I told you, is that right now the primary open angle glaucoma is a complex disease, and more than 25 loci are there. And then in addition to the main 25 genes, see, there are, it is associated with the other genes. See, the genes, reported to associate with the POG, not only the main candidate genes, it is also associated with the other genes. The pathway, see here, the pathway, this is the complex pathway. That's what, more than 25 genes, 25 loci, and other genes are associated. It seems to be, it's a very, very complex. I think for your information, very recently, last month, there is a paper from Spain. They published the paper in molecular evolution concerned with the they targeted with the cancer. So I think the human gene, I think if you see I think when the human genome project was completed, they told 30 to 40,000 genes. Now the number is reduced, keep on reducing. It means right now the recent report is the human genome, the number of genes in human genome is only 19,000. Only 19,000. Okay, it means they correlated exactly the protein. Whatever through the proteomic approach, they identified only 19,000 genes are there in human genome. And then, so this is the review. If you are interested, you can go through. All the informations are there. This is a very recent review from Rand Allingham. And then. So this is the entire summary of the GWAS. The people, the genome-wide association studies. And then it's very interestingly, we are focusing on one particular gene in our lab right now. So I think, see, so here there is a SAX6, this is one of the gene, say SAX6 gene. I think we are working on this particular gene right now in our lab. Um, Okay, so in addition, I think I told, as I mentioned, myocin is the main candidate gene. So if you see at least 100 patients, primary open angle glaucoma patients, any population, the prevalence is 2 to 4%. And even when we screened, a little bit higher, we got 8%, 6 to 8% in our Indian population. And then the WTR36 is not that much, association. I think they reported from US, but that is not, the right candidate gene. That is concerned with the Indian population. And the optinurine, again, this is I think here it is not a specific gene for only for the primary open angle glaucoma. It is also, candidate gene for normal tension. If we screen like 100 patients, like 15% of them primary open angle glaucoma, 10% of them, normal tension glaucoma. Again, this is not a specific gene for primary open angle glaucoma. And so I think what we did, there is a multicentric project between Aravind, Sundaresan, LV Prasad, as well as from Kunal Ray group from Calcutta and AIMS. So I think that time, long back, it's like 8 or 10 years ago, we are highly excited. Why not we make a chip for diagnosis, especially for the Indian population for primary open angle glaucoma? That's what we want to make, a multicentric thing, and then make a chip for primary open glaucoma, collect more than 2,000 samples in all the 4 centers. So here, I think this is my second PhD student work. This is the myocin gene, and then when we screen, this is particular the exon 3. This olfactory domain. This is a caught spot, and then more mutation you can identify here in all the population. So we did, as I mentioned, we identified 6 to 8% of mutation in this particular region. So I think this is the overall picture of the world, and here I think what we compiled all the mutations identified in myocin and our part in the red. So that kind of mutations we well documented in Indian population. In addition, this is these 7 mutations we identified. And then you back to the second point I mentioned, because of the extracellular matrix is they increase the pressure. So there is a mutation called glycine to 367 arginine. This is, when compared to the case, you take all the 1,000 cases of the POAG, one particular case, it is very big. It means that he is keep on coming to the hospital. And when we see the entire pedigree, and then we collected all the sample, we analyzed the father, mother, and then the younger and elder brother. The elder brother is right now, when the diagnosis is around 45, and then he coming to the hospital for 10 years, 55. So I think when we analyzed the whole family, so we identified even the younger brother, he is 125, but we identify the same mutation in the family. And then, so the thing is, the onset of the disease, you know the classification better than me. If it is a juvenile, is more than 35. If it is more than 35 or 40, that is a primary open angle glaucoma. That particular patient is prone to develop for primary open glaucoma. He is working as a conductor in Madurai. So and then we educated. I think you better to check your eye and pressure, and then you come to the hospital regularly whether it is you are going to prone for the disease. So like that counseling we are giving to the many of the families, as well as we proved yes, this is this is an excellent mutation, and functionally we proved yes. This is associated with the disease by cloning and all the gene expression studies. This particular thing in our lab. And then again, as I mentioned, these are the optinurine genes and WTR36, and these are the mutations we have identified in our lab. And then, so I think this is the multi-centric study at the 4 centers. So we had all the 4 centers from LVP and Aravind, Sankaranidralaya and Kunal Ray group and the AIMS. So we identified more than 1,000 samples, and then only the myocin, what is the myocin mutation in our population, as well as. And there is another candidate gene, this is called SIFON1B1 gene. This is the gene for congenital glaucoma. It is having a kind of combining effect with these 2 genes. So we also analyzed, even though it is the candidate gene for congenital glaucoma, so we analyzed for primary open angle, and then we identified the mutations, and then we correlated with the disease. So like that, ultimately, our ultimate aim is to make a DNA chip for diagnosis. The problem when we pool the sample, I think the ethnicity variation. Suppose some of the hospital in the cosmopolitan city, and Madurai is in a remote place surrounded by villages. So like that, we can't be able to get a proper, I mean, one particular marker here. I'm sorry, no, I'll rush up. And in addition, I mean, there is a pseudo exfoliation. I think this is another, I mean, the secondary complications. And so there is a gene called LOXL1. So this particular gene, we also, I mean, we have taken the sample like a pseudo exfoliation alone, and the pseudo exfoliation with the glaucoma, and then control samples this particular LOXL1 gene, and then we proved, I mean, more than 500 samples is associated with the primary this pseudo exfoliation. When Western population, I think these are the genes. I mean, Gerald is asking to move the slides. I will do that. Okay, I think this is a very recent finding. And this is one of the landmark publication recently, day before yesterday, published in Nature Genetics. So we screened more than 20,000 samples from 17 countries, from 6 continents. The Ongtin group and our clinician, Dr. Haripriya and everything, we published that. But this paper is published in Nature Genetics, and this is another new candidate gene for pseudo exfoliation. And these are the recommendations. Please, I mean, if you are, I don't have time, or I can interact with you, I mean, today or tomorrow. I think some of the labs, like Dr. Edwin Stone lab and Jenny Wicks from Boston, and then, based on this, whatever the finding for the past, 20 years, they are recommending, like if it is a congenital glaucoma with the family history, you can go for, this particular gene. Or if it is like that, they are giving some recommendation. Based on that, definitely, if the patient is interested, we can go for, the screening. Okay, so then I think this is another landmark study. I think Dr. Kumara Manikvel and group, I think they published a paper from angle closure glaucoma. Even though the Human Genome Project is completed, the candidate gene for angle closure is not angle closure glaucoma. I'm switching the gear to the angle closure. So here, angle closure, the candidate gene is not identified for more than 10 or 15 years. So now, very recently, Dr. Ongtin and Kumara Manikvel contributed excellent, many contribution for their group in Sankar Nitralaya, and they identified the gene. And so I think we can't able to join in that particular discovery or the replication. So once the paper is published, my student is here, my 17th PhD student right now, she's sitting in the crowd, and sees the Rupam. So I think she's a very smart, PhD student, and we analyzed more than 500 samples within 1 month period, and then is one of them. This paper is, we replicated the study, and then we published in IOVS. And another recent, thing is, so I think in addition to the, right now, you can approach either in the DNA or the end product of the protein. So here, what right now, our institution is the Center of Excellence for Proteomics for fungal keratitis and other thing. So right now, I think this is another area you can deal with in addition to the DNA, you can deal with in the proteins. The protein, in the sense, you can take any biological fluid. If it is keratitis, you can take tear. If it is POAG, you can take serum or aqueous humor. You take that the biological fluid, you separate the protein as a two-dimensional electrophoresis, and then you can compare the glaucomatous and non-glaucomatous, and then separate the protein, and then you can, if there is any difference between the two, the control and diseased, and then you pick up the protein and then put in the LCMS. It means in high pressure, that the protein converted into ions, it will pass through the channel, and then it will give a mark. Based on that particular technique, we can able to identify that particular, any markers or the gene is associated with the primary operatic glaucoma. The same way here, instead of targeting the DNA, right now people are targeting a specific tissue. You better to target suppose any tissue, even if it's a retina or AMD, anything, you better to pick up exactly the affected tissue. If it is a retina or AMD, you sometimes the biopsy is possible, sometimes it is impossible. So glaucomatous thing, we can able to get the tubercular meshwork during the tuberculotomy. So I think, we are tired, in the sense, you can't get the pure TM. When you grow in the medium, I think the scleral cells, it will dominate, and then you will get the mixed population. So like that, right now, we are targeting, we are classifying the cadaverized, whether it is a glaucomatous or non-glaucomatous. Based on that, we are getting the TM, and then we are working on that. So here, based on this, people are working on the gene expression studies. Based on the gene expression studies, definitely we can able to say, yes, this particular gene is upregulating or downregulating in the disease process, and then we can make we can correlate with the pathogenesis. Sorry, Gerald, I'm sorry. I think I'm speaking too fast, but even I can't able to complete. I will do that. This is the thing. And this is the some of the, as I mentioned, the University of Iowa having the thing. And there is another one interesting thing, already Gerald, talked about. This is from the Duke University. I think, obviously, ultimately, you have to, make how to take the research from bench to bedside. So before that, you have to prove in the animal model. So they have taken the particular gene, and then they cloned the GFE, the green fluorescent protein, and then they inject it into the mouse, and then they are checking the expression. See here. So the expression of the TM is in the cornea as well as in the meshwork. It is not in another place. It means this is the right model. They justified. They use the lentiviruses. Usually people use adeno-associated virus. Even some people are. That's what Gerald put a slide about from Alkan lab. So he is also working on. I'm sorry. So this is the adeno-associated virus related to the function of they proved the expression is there. Okay. And this is again, if you are interested in the gene therapy, I think they are making attempt. They are making different viruses and everything for the therapy. So that is even though it is not successful, people are working on that. And so that's what I'm telling. So we are doing the proteomic approach. So not only for the glaucoma, so diabetic retinopathy, fungal keratitis, and everything, we are doing that one. And I think this is this one or two slides. Gerald, please forgive me. And this is the very recent report from Scientific American. So I think the Human Genome Project, like previously $5,000 you can able to sequence the entire genome of a particular person. Now they slash the price to 1,000, even that the dollar amount is going down if you increase the sample size. So I think, suppose a researcher point of view, I can able to say, I can recommend, yes, the entire sequence is needed, the entire genome is needed for to study the particular patients. But here, I think you see here, the bottom line is the scientists are ready to, for the 1,000 genome, but the government and the medical system, doctors and the general public are almost, certainly not. So I think, this is we see, without, clinician, we can't able to do research. That's what is the last slide. I think, so I think when you want to study a particular, any disease, not only glaucoma, so you better to interact with the researcher, and then you give a exact, sample size for research. So that's what I'm putting here. Thank you, John. Gerald, I'm sorry.